Hello friends, welcome to Topper's Tips. Today I am going to complete this chapter cell cycle and cell division in 10 minutes. You can just go through this video and gonna get the most important points in this chapter. So let's start with it. So what's cell cycle? It is the sequence of events by which a cell duplicates its genome, synthesizes the other constituent of the cell and eventually divides into two daughter cells. This is what the cell cycle is. Now, although the cell growth it is a continuous process, DNA synthesis occurs only during one specific stage in the cycle, and that is the synthetic phase. Now we are going to come to that later. Now, when we talk about the phases of cell cycle, so first of all, the cell divides once the human cell basically divides once after 24 hours. And if we talk about yeast cell, it takes 90 minutes. The cell cycle it takes just 90 minutes for one cycle to complete. Now there are two basic phases: interphase and M phase. The M phase is when the actual cell division occurs, and interphase is the phase between the two successive M phases. So the cell division it mainly occurs for 1 hour. From the 24-hour टाइम पीरियड इट अकर्स ओनली फॉर 1 hour. एक ही घंटे के लिए होता है. एंड इंटरफेस it remains for most of the time, 95% of the duration of the cell cycle. Now M phase starts with nuclear division and then the separation of the daughter chromosomes, and then we have the division of the cytoplasm, that is cytokinesis. Now the interphase, we call it the resting phase, and it is the time during which the cell is basically preparing the cell growth and DNA replication is occurring, and the actual cell division is not occurring during this stage. The interphase is divided into three phases: G1, S, and G2 phase. The G1 phase corresponds to the interval between mitosis and initiation of the DNA replication, and it is the metabolically active, and it continuously grows, but the cell continuously grows, but it does not replicate its DNA replication होता है आपकी synthesis phase में, and there is no increase in the chromosome number. When we talk about the DNA synthesis, only the DNA amount doubles and not the chromosome number. The chromosome numbers remains the same. That's an important point. And S phase में we also have the centriole duplication which occurs in the cytoplasm. G2 phase, we have the protein synthesized in the preparation for mitosis, where the cell growth continues. Now there are some cells that do not divide continuously. They exit the G1 phase and enter into an inactive stage. So हम quiescent stage बोलते हैं of the cell cycle. Cells in this stage, they are metabolically active, but they don't proliferate unless they are asked to do so. Mitotic cell division, वैसे तो we just see in diploid somatic cells, but few exceptions we have where haploid cells also divide like the male honeybees. And talk about the M phase. Now this is the most dramatic period. Why the most dramatic period of the cell cycle? Because it involves major reorganization of virtually all components of the cell, and the number of chromosomes here we have in the parent and the progeny remains the same, so it's equational division. When we talk about karyokinesis, it's divided into 4 stages: prophase, metaphase, anaphase, and telophase. Prophase, it is the initial. If we talk about the initiation of the prophase, we have the condensation of the chromosomal material, and it becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during the S phase, now begins to move towards the opposite poles of the cell. So if we talk about the main characteristic events of prophase, so the chromosomal material it condenses, का condensation हो जाता है, compact mitotic chromosomes बन जाते हैं, and they are composed of 2 chromatids attached together by the centromere. Centrosome जिसने duplication during the interphase किया था, these centrosomes they move towards the opposite poles, and they radiate the microtubules asters, and these asters with the spindle fibers ये mitotic apparatus बनाते हैं. Now cells at the end of prophase हम देखते हैं तो Golgi complex, endoplasmic reticulum, nucleolus and nuclear envelope इसमें से disintegrate हो जाता है, disappear हो जाता है. When we talk about metaphase, complete disintegration of the nuclear envelope is the start of the second phase. Now here the condensation of chromosomes is completed. इस स्टेज में हम morphology of the chromosomes को easily study करते हैं, and metaphase chromosome is made up of 2 sister chromatids, which are held together by the centromere, and we have small disc-like structures at the surface of centromeres जिसको हम kinetochores बोलते हैं, and these structures they are the attachment site for the spindle fibers that जो कि सेंट्रियोल से जो स्पिंडल फाइबर्स जो माइटोटिक अपैरेटस था उसके अटैचमेंट साइट है आपके kinetochores. Metaphase में chromosomes they lie at they come and form an metaphasic plate. Metaphasic plate क्या है कि they alignment of the chromosomes at the metaphase it is referred to as metaphase plate. अब इसकी जो की फीचर्स हैं, की फीचर्स ऑफ मेटाफेज इज दैट स्पिंडल फाइबर्स kinetochores को अटैच हो जाते हैं एंड द क्रोमोसोम्स दे मूव टू द इक्वेटर एंड दे गेट अलाइंड अलोंग द प्लेट and they form the metaphasic plate. This kind of a plate is the metaphasic plate. Now we have anaphase. Now anaphase, each chromosome arranged at the metaphase plate is split and two daughter chromatids or referred as the daughter chromosomes or the future daughter nuclei. They begin the migration towards the opposite poles. Now each chromosome moves away from the equatorial plate and the centromere of each chromosome remain directed towards the pole. वो pole की तरफ खींच रहे हैं जो mitotic apparatus थे, they are trying to pull the centromere of each chromosome remains directed towards the pole and hence the leading edge with the arms of the chromosome trailing behind. So basically वो ऊपर की तरफ खींचते हैं towards the poles. So basically the key events of anaphase is कि centromere split हो जाते हैं, chromatids separate हो जाते हैं, and these chromatids they move towards the pole की तरफ उनको खींचा जाता है with those mitotic apparatus. Now let's come to the fourth and the final stage of karyokinesis that we have telophase. Chromosomes अब उनके respective poles पे पहुंच चुके हैं and they decondense. And this stage shows the events जो उसके key features हैं कि chromosomes cluster at the opposite spindle poles and identity is lost. Now they form the chromatin material again वो बन जाते हैं chromosomes जो पहले कॉम्पैक्ट थे they again become that chromatin material. न्यूक्लियर एनवेलप और जितने भी न्यूक्लियोलस गोल्जी कॉम्प्लेक्स वे सारे वापस आ जाते हैं. इट्स जस्ट लाइक द अपोजिट व्हाट हैपेंड ड्यूरिंग प्रोफेज. लेट्स टॉक अबाउट साइटोकाइनेसिस. सो divided into two daughter cells by cytoplasm called cytokinesis, cell division gets completed. Now plant and animal cells में it's different. In animal cell it is achieved by the appearance of furrow in the plasma membrane. Furrow deepens and ultimately center की तरफ जो है वो furrow बाहर से center की तरफ बनता है and then the cytoplasm gets split into two. But plant cell में because it has an inextensible cell wall, therefore it undergoes cytokinesis by different mechanism. Formation center से चालू होता है and goes outwards to meet the lateral walls. So new wall begins with the formation of precursor cell plate का formation होता है which represents the middle lamella between the walls of two adjacent cells. Now at the time of cytoplasmic division, organelles like mitochondria and plastids they also get distributed between the daughter cells. Now in some organisms karyokinesis is not followed by cytokinesis. तो क्या हो जाएगा, एक सेल के अंदर बहुत सारे न्यूक्लियस हो जाएंगे, साइटोप्लाज्म स्प्लिट नहीं हुआ है, न्यूक्लियस बढ़ चुके हैं. So it creates a multinucleate condition which we call the syncytium and it happens in liquid endosperm in coconut. Let's talk about the significance of mitosis. Is usually restricted to diploid cells only, but some lower plants and some social insects they also divide by mitosis. It results in the production of diploid daughter cells with identical genetic component. Genetic component identical रहता है and the growth of multicellular organisms it is also due to mitosis. Cell growth में जो disturbance से जो disturbance होती है रेशियो में न्यूक्लियस और साइटोप्लाज्म की वो रिस्टोर कर देता है माइटोसिस. And the cells of the upper layer of the epidermis and the cells of gut, blood cells जो कि constantly replace होते हैं या फिर meristematic tissues, apical and lateral cambium result in the continuous growth of the plants throughout the life. So mitosis इन सब में help करता है. Now we talk about meiosis. When we talk about the production of offspring by sexual reproduction includes the fusion of two gametes. So इसमें complete haploid set of chromosomes. And we need the haploid set of chromosomes तो किससे meiosis से होता है. So chromosome number It's reduction division where the chromosome number reduces to half. Now meiosis में 2 sequential cycles of nuclear and cell division होते हैं, but remember it's single cycle of DNA replication. Meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids at S phase. Now meiosis involves the pairing of homologous chromosomes and recombination between the non-sister chromatids. homologous chromosomes कि जो नॉन सिस्टर क्रोमैटिड्स हैं उनके बीच में it results in the pairing of the in that. Now 4 haploid cells is not like mitosis. यहाँ पे 2 haploid cells after meiosis 2 बनते हैं. Now meiosis 1 में हम जब prophase देखें so it's a bit longer and complex. Prophase 1 itself is divided into prophase 1 itself is divided into leptotene, zygotene, pachytene and diplotene stage. So when we talk about the leptotene stage, chromosomes they become visible under the light microscope and the compaction of the chromosome continues throughout the leptotene. जब हम बात करते हैं लेप्टोटीन को वी ऑल्सो कॉल एज बुकेट स्टेज, क्योंकि वो उस तरीके से आपके जो है क्रोमोसोम्स दे स्टार्ट बिकम द क्रोमैटिन दे स्टार्ट बिकम कॉम्पैक्ट जस्ट स्टार्ट ही होता है. एंड साइगोटीन में द क्रोमोसोम्स दे स्टार्ट पेयरिंग टुगेदर, इसको हम साइनाप्सिस बोलते हैं. एंड सच पेयर क्रोमोसोम्स आर होमोलोगस क्रोमोसोम्स. नाउ यहाँ पे क्रोमोसोम साइनाप्सिस इज एक्कम्पनीड बाय फॉर्मेशन ऑफ कॉम्प्लेक्स स्ट्रक्चर. इस कॉम्प्लेक्स स्ट्रक्चर को हम साइनैप्टोनिमल कॉम्प्लेक्स बोलते हैं. द कॉम्प्लेक्स फॉर्म बाय द पेयर ऑफ साइनाप्स्ड होमोलोगस क्रोमोसोम्स उसको हम बाइवैलेंट या फिर टेट्राड बोलते हैं. ये नेक्स्ट स्टेज में ज़्यादा क्लियर दिख सकता है. फर्स्ट 2 स्टेजेस जो होते हैं थोड़े शॉर्ट लिव्ड होते हैं. टॉक अबाउट द थर्ड स्टेज जो कि पैचीटीन है, उसमें 4 क्रोमैटिड्स ऑफ ईच बाइवैलेंट क्रोमोसोम्स बिकम डिस्टिंक्ट एंड अपीयर एज टेट्राड. This stage is characterized by the appearance of recombination nodules. The site at which the crossing over occurs between 2 non-sister chromatids of homologous chromosomes. The crossing over क्या है? It is the exchange of the genetic material between 2 homologous chromosomes. It is also crossing over एक enzyme mediated process है. enzyme होता है recombinase and recombination of the genetic material of the 2 chromosomes इसमें होता है. recombination between the homologous chromosomes end of पैचीटीन में ख़त्म हो जाता है. By the end of पैचीटीन it's finished and leaving the chromosomes linked at the sites of crossing over. डिप्लोटीन में dissolution of the synaptonimal complex होता है. जो recombined homologous chromosomes of the bivalence है वो separate होना चाहते हैं except the site of crossover. crossover जो जहाँ पे वो अभी सेपरेट नहीं हुए हैं उसको एक्स शेप्ड स्ट्रक्चर्स बोलते हैं. वी कॉल इट चियास्माटा. और उन साइड्स ऑफ सम वर्टिब्रेट्स ये जो डिप्लोटीन स्टेज होता है ये ज़्यादा टाइम के लिए इट कैन लास्ट फॉर मंथ्स और ईयर्स और हम इसको डिक्टियोटीन स्टेज बोलते हैं. डिक्टियोटीन स्टेज. अब जो फाइनल स्टेज होता है उसको हम डाइकाइनेसिस बोलते हैं. इसमें टर्मिनलाइजेशन ऑफ द चियास्माटा भी टर्मिनलाइज हो जाता है. क्रोमोसोम्स फुली कंडेंस्ड हो जाते हैं और जो मियोटिक स्पिंडल होता है वो हम असेंबल हो जाता है फॉर टू प्रिपेयर द होमोलोगस क्रोमोसोम्स फॉर सेपरेशन. By the end of dikinesis, न्यूक्लियोलस डिसअपियर, न्यूक्लियर एनवेलप ब्रेक डाउन और फिर आपका मेटाफेज चालू हो जाता है. ना मेटाफेज जो बाइवैलेंट क्रोमोसोम्स थे अगेन अलाइन ऑन द इक्वेटोरियल प्लेट एंड वो मेटाफेजिक प्लेट बना देते हैं. माइक्रोट्यूब्यूल्स अपोजिट पोल्स ऑफ द स्पिंडल अटैच टू द काइनेटिक कोर उसको पुल करने के लिए टुवर्ड्स द पोल्स. ना एनाफेज में वैसे ही होमोलोगस क्रोमोसोम्स जो सेपरेट होते हैं सिस्टर क्रोमैटिड्स रिमेन एसोसिएटेड एट देयर centromeres. Nuclear membrane. Now if we talk about telophase 1 तो न्यूक्लियर मेम्ब्रेन और न्यूक्लियोलस जो है रीअपियर हो जाते हैं. साइटोकाइनेसिस फॉलो होता है एंड दिस इज नोन एज डायड ऑफ द सेल्स. डायड बनाते हैं. दिस स्टेज बिटवीन 2 mitotic divisions is interkinesis and it is generally short-lived. There is no replication of DNA during interkinesis. जब 2 meiotic divisions के बीच में स्टेज होता है वी कॉल इट इंटरकाइनेसिस एंड इट इज़ शॉर्ट लिव्ड. वहाँ पे कोई भी डीएनए रेप्लिकेशन नहीं होता है. एंड आफ्टर दैट वी हैव प्रोफेज 2 जो कि सिंपल होता है. देन प्रोफेज 1. इफ आई टॉक अबाउट मियोसिस 2, सो मियोसिस 2 इनिशिएट में हम कर सकते हैं साइटोकाइनेसिस And before chromosomes fully elongate करने का time मिले, meiosis too resembles normal mitosis. Mitosis के काफ़ी it's just like the mitosis, and nuclear membrane disappears by the end of prophase 2. Chromosomes again become compact. Now at this stage, when we talk about metaphase 2, chromosomes again align at the equator, and microtubules from the opposite poles get attached to the kinetic cores of the sister chromatids. Anaphase 2 में क्या होता है? Again, splitting of the centromere of each chromosome, and the opposite जो mitotic apparatus है, it pulls the chromatids towards itself, or it moves towards the poles. When we talk about now telophase 2 is the end of meiosis, and two groups of chromosomes, they again get enclosed by the nuclear envelope. Cytokinesis follow in the formation of tetrad of cells, and 4 haploid daughter cells जो है formed हो जाते हैं. When we talk about significance of meiosis, so meiosis is the mechanism in which conservation of the specific chromosome number कर सकते हैं. Each species की we are able to maintain that chromosome number of each species जो कि haploid हो जाता है in sexually reproducing organisms, and again it is back into the species after fertilization. So it results in the reduction of chromosome by half. It increases the genetic variability in the population of the organism, or variations. You are all aware of that variations are important for the process of evolution. So guys, here we have completed the chapter. जितने important points हो सकते थे I have covered in these 10 to 15 minutes. Hope you like it. Do share, like and subscribe. Thank you so much.